Outbreeding devices. Majority of flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flower. Continuous self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of pollen. In some other species, the anther and the stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both the devices prevent autogamy. The third device is to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self-pollen from the same flower or other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize monoecious, it prevents autogamy but not zoonogamy. In several species such as papaya, male and female flowers are present on different plants. That is, each plant is either male or female dioicy. This condition prevents both autogamy and zoonogamy. Pollen pistil interaction. Pollination does not guarantee the transfer of the right type of pollen in compatible pollen of the same species as the stigma. Often pollen of the wrong type, either from the other species or from the same plant, if it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of the right type, compatible, or of the wrong type, incompatible. If it is of the right type, the pistil accepts the pollen and promotes post-pollination events that leads to the fertilization. If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grain and the pistil. This dialogue is mediated by chemical components of the pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interactions leading to the recognition followed by acceptance or rejection. As mentioned earlier, following compatible pollination, the pollen grain germinate on the stigma to produce a pollen tube through one of the germ pores. The contents of the pollen grain move into the pollen tube. Pollen tube grows through the tissues of the stigma and style and reaches the ovary. You would recall that in some plants, pollen grains are said at two-celled condition, a vegetative cell and a generative cell. In some plants, the generative cell divides and forms the two male gametes during the growth of pollen tube in the stigma. In plants which celled pollen in the three-celled condition, pollen tubes carry the two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergies through the filiform apparatus. Many recent studies have shown that filiform apparatus present at the micropylar part of the synergies guides the entry of pollen tube. All these events from pollen deposition on the stigma until pollen tubes enter the ovule are together referred to as pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeder in manipulating pollen pistil interaction, even in incompatible pollination, to get desired hybrids. You can usually study pollen germination by dusting some pollen from flowers such as pea, chickpea, crotalaria, balsam, and vinca on a glass slide containing a drop of sugar solution, about 10%. After about 15 to 30 minutes, observe the slide under the low power lens of the microscope. You are likely to see pollen tubes coming out of the pollen grains. A breeder is interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiments, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination from unwanted pollen. This is achieved by emasculation and bagging techniques. If the female parent bears bisexual flowers, removal of anthers from the flower bud before the anther dehiscence using a pair of forceps is necessary. This step is referred to as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. This process is called bagging. When the stigma of bagged flower attain receptivity, mature pollen grains collected from anther of the male parent are dusted on the stigma, and the flowers are rebagged and the fruits allowed to develop. If the female parent produces unisexual flower, there is no need for emasculation. The female flower buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower rebagged. 1.3 Double fertilization. After entering one of the synergies, the pollen tube releases the two male gametes into the cytoplasm of the synergid. One of the male gametes moves toward the egg cell and fuses with its nucleus, thus completing the syngamy. This results in the formation of a diploid cell, the zygote. The other male gamete moves toward two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus (PEN). As this involves the fusion of three haploid nuclei is termed triple fusion. Since two types of fusion, syngamy and triple fusion, take place in an embryo sac, the phenomena is termed double fertilization, an event unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell (PEC) and develop into the endosperm, while the zygote develops into an embryo.